Hello, good morning, friends. I'm presenting you the topics about the cancer and the genetics. What are the relation between the cancer and then there's a hereditary component and then a familial cancers. So we'll be addressing about you in this short presentation and then a slide showing to you proto-oncogene and an oncogene. The concept must be very clear that the proto-oncogene is present in every individual, but unless until it get mutated and become an oncogene, it's not harmful. It's not a harmful situation. So that the oncogene is responsible for the maximum amount of a damage and a development of a cancer in the individual. So myself, Major Dr. M.Q. Baig, presenting you the topic cancer and genetics. So what are the causative agents which converts this DNA, the proto-oncogene, into oncogene? We must know this thing. The proto-oncogene in this figure you see here, the proto-oncogene is there, but it is not. It's harmless unless until it gets some external agents or internal agents to disturb this their nucleotide sequences, like the radiation, like carcinogenic chemicals, even the chemotherapy, ultraviolet radiation, mutations. These lead to the proto-oncogene into oncogene, and then oncogene, that cells start behaving like a cancer cells with a rapid proliferation and uncontrolled proliferation. So, so far as the estimated uh, data, 5 to 10% of all cancers have hereditary components. Among the total cancers, the 5 to 10% having a definite hereditary component. Then there are over 35 hereditary cancer syndrome, basically cancer susceptibility syndromes, many with overlapping phenotypes. Maybe different phenotypes are there and causing the hereditary susceptibility syndrome, which will be discussed in the further slide. The how in this slide you see here, the how the normal cells becomes a cancer cells, the normal cells, the DNA mutation and the uncontrolled proliferations. And then I've told you the DNA mutation, the causative agents may be hereditary, may be radiation, chemical, or spontaneous mutations during the DNA duplication. This lead to the cancer, particularly in the hereditary form of cancer. So this is again a Robertson translocation. You see here the two chromosomes, and then after the translocation, two chromosomes stick together. Suppose the blue chromosomes, which is shown in the figure, is having an oncogene and is stick to the uh, that other orange colored showing the chromosome, so that the transformation gone into the this chromosomes is becomes a altered chromosomes and may lead to the cancer. So there are different type of translocation is there in the mutation and during the cells replications, which may lead to the cancer. Now regarding the next generation sequencing, which is a real hope for gene sequencing and identifying the group of a population or an individual that they are having a strong component of development of a cancer. So these next generation sequencing is basically we have a lot of damage with the COVID-19. But what happens in the COVID-19, these technology, next generation sequencing, AGS, is got a massive uphold regarding the development of a lab, regarding the development equipment, and regarding the expertise of individuals, professionals to think of a next generation sequencing because in a very short time, so it has got a boost. The COVID has damaged so much of human population, but also given a ray of hope for development of next generation sequencing. So basically, next generation sequencing is a basically massively parallel or a deep sequencing related term that describe a DNA sequence which has revolutionized the genomic research. Using the next generation sequencing, an entire human genome can be sequenced within a single day. When earlier, in contrast, previous Sanger sequencing technology used to require over a decade of a time to deliver the same result. So it is a very fast technology which can sequence the human genome on a very fast track. Though this technology is growing very fast, but yet it is not being translated into the routine oncology clinical practice all over the world. I'm telling you the word all over the world because not India is only suffering with the problem of development of a next generation sequencing lab, particularly for the use of oncology. Now, the few private stakeholders have indulged in this new branch for development of next generation sequencing and then a genetic analysis of a tumor tissue, particularly biopsy tissue. They use for this next generation sequencing. The knowledge gained through the use of next generation sequencing is increasing our understanding of the natural history of hereditary cancer syndromes and contributing and expanding the phenotype associated with individual cancer susceptibility genes. Also, next generation sequences, this decrease the time to a potential diagnosis and reduce testing fatigue for a patient's family and providers. So they cut short the time. You can cut a very short time. It can give a definite diagnosis of a multiple hereditary cancer syndromes. The best panel for a given clinical situation should be selected. I mean, what it does means? It means that we should select a very important responsible gene for a real development of a cancer, and that genes should be considered for a panel of examination by NGS. Including Actually, if you include a panel of low penetrance or newly identified genes without a very specific action and ability, may be a problematic for interpretation of results. So that is the importance of this thing. There is a use of a genetic counselors. They are an essential part of hereditary cancer risk assessment team, helping the medical team select the most appropriate test and to interpret the often complex result. The counselors also help in assisting the testing of a family members at a risk. Counselors obtain an extended family history, counsel of patients on available test and potential implication of a result for themselves and their family members. And then a counselors also explain to the patient the implication of a test result, post test counseling. So these are the advantage if you go through the proper chain of involving the genetic counselors for the encouraging the patients and their relative and then volunteers for the genetic testing by NGS technology. The genetic testing for cancer susceptibility should be offered when the following conditions are met. It's not that we can randomly design this thing that we randomly do testing of a NGS. It should be have some criteria, some guidelines must be followed. First, the individual has a personal or family history suggestive of hereditary cancer syndrome. Second, the test result can be adequately interpreted. The test result will influence the medical management of individual and their family members. Third point, the fourth point is testing accompanied by the informed consent. It should be a taken informed consent for conducting the genetic test. So the risk assessment tool exists to estimate the likelihood that the patient may carry a mutation in a particular cancer susceptibility genes. There is no absolute threshold to consider the genetic testing. This line is very important. Till now, there is no absolute threshold for considering the genetic testing for individual. When the risk models can be used as a guidelines, it is the clinical judgment of oncology and genetics care providers that they should determine the appropriateness of genetic testing in a given situation. So the both the molecular oncologist and clinical oncologist, medical oncologist, say the both a team of a member should decide that the patient has to go or individual has to go for a genetic testing or not. The individual undergoing testing, the recommendations are made. The moderate penetrate genes confirm a lifetime cancer risk for 20 to 50% or 2 to 4-fold increase above the general population risk. So even the moderate penetrance of a genes or alteration of genes actually increases the cancer risk in compared to the normal individual. In that individual, 20 to 50% or 2 to 4-fold high. Low or unknown penetrance genes may have a limited or conflicting evidence, providing the example of gene that may be included in the each group. So the low penetrance genes or unknown penetrance genes should not be the criteria to do the genetic testing and genes testing. So regarding the few cancers like a breast cancer and ovarian cancer, the referrals, the hereditary breast and ovarian cancer syndrome are among the most common indications seen in the cancer genetics clinic. The hereditary breast and ovarian cancer syndrome is caused by basically the mutation of BRCA1, breast cancer antigen 1 and breast cancer antigen 2, BRCA1 and BRCA2, and remains the primary consideration for an individual with a personal or a family history of early onset breast cancer. So where we should consider is the multiple relatives with the breast cancer, male breast cancer, breast and ovarian cancer in the same lineage. The current prevalence estimate for BRCA1 mutation in 1 in 300 and for BRCA2 is 1 in 80010. So that it shows the BRCA1 mutation is more common in compared to the BRCA2 mutation. So mutations in the BRCA1 and BRCA2 are associated with the increased risk of early onset breast cancer, multiple primary breast cancer, epithelial ovarian cancer, primary peritoneal cancer, male breast cancer, risk for melanoma, risk for prostate, risk for pancreatic cancers. So that BRCA1, BRCA2 is not limited up to the breast cancer. You see here the gambit of cancers are associated with this. So BRCA1 mutations ranges from 46 to 65% in usually, and a BRCA2 mutation is 43 to 45%. So almost 45% you can consider in a woman with the pooled analysis data, which is shown. So the BRCA1 mutation, I have already told you, it's a very common mutation. It's high mutation range is high in compared to BRCA2 mutation. So where are your BRCA1 and BRCA2 basically? The chromosome 70 have a BRCA1 gene and a chromosome 13 having a BRCA2 gene. So one should remember where these where these genes are lying in the chromosome. So the breast cancer with triple negative features like estrogen, ER, and HER2 all are negatives. They are associated with increased risk of BRCA1 gene mutation. Though the BRCA2 mutations have also been found in the women with triple negative cancer, A woman with ovarian cancer have 13 to 18% chances to have a BRCA gene mutation. Sometimes isolated ovarian cancers, approximately 15% of women with invasive ovarian cancer have detectable mutations in BRCA1 or BRCA2. Testing for BRCA1 and BRCA2 gene is recommended for all women diagnosed with ovarian cancer. So, what are the role of genetic analysis for gastrointestinal tumor? Multi-gene cancer panel have a greatest clinical utility in setting of a gastrointestinal cancer. History where phenotype overlap is significant and differential diagnosis include multiple gene. There it is very much indicated. And up to 6% of colorectal cancer may be due to definite high risk syndrome, Lynch syndrome, and have alone having a 5 gene EPCAM, MLH1, MSH2, MSH6, PMS2. So they are the 5 genes in the Lynch syndromes have been identified, and the 6% of colorectal cancer may be due to the definite hereditary component. So, tumor screening with immunohistochemistry studies may identify these genes to target, but does not definitely rule out the role of evolution of all 5 genes. The NGS panel may now be preferred as a frontline evaluation for Lynch syndrome in event that ISC testing is unavailable. So, the pancreatic cancer, approximately 5 to 10% pancreatic cancers. Almost we know the histology is more common of adenocarcinoma is familial in tendency, and if 5 to 10% pancreatic cancers are familiar in tendency, and the several genes have been associated increased pancreatic risk cancers, the BRCA1, BRCA2, CDKN2A, PLAB2, PRSS1, STK11, Lynch syndrome genes. These are the genes responsible for pancreatic cancer as well. So, the American College of Gastroenterologists currently recommend the testing for BRCA1, BRCA2, PLAB2, CDKN2A, and ATM for all familial pancreatic cancer syndromes or pancreatic cancers. Adding the genes for Peutz-Jeghers syndrome, Lynch syndrome, hereditary pancreatitis must be recommended for NGS testing if there is a clinical history suggestive of these conditions. So that we know that the importance of next generation sequencing is very, very important. And then one should know about the role of NGS in the cancer, and because of a fastest result and a more accurate results. But there are some criteria to follow regarding the next generation sequencing. Thank you very much, my dear friends. Wish you have a good day. Thank you.